Ashevcu from the Institute for Nanomaterials, Advanced Technologies and Innovations of the Technical University. And it's my pleasure to invite today Dr. Kolis Audach, who is going to give a talk on the efficacy and toxicity of nanoencapsulation of natural products for drug delivery purpose. Dr. Kolis was a postdoc at the Yale University in the United States, and now he is a director of academic research and community services at the Swiss-German University in Indonesia. And maybe I can just briefly introduce his field. He works now for exploration of Indonesian biodiversity for drug discovery through development of extract library focusing on antimicrobial agents, or he is dealing with the investigation of drug-resistant bacteria or emerging pathogens by using metagenomics approach for diagnostics or early detection of particular diseases. So again, I'm very happy I can invite Dr. Kolis Audach. And Kolis, maybe you can start. The floor is yours. Thank you.
Okay, thank you very much, Anna. Very good afternoon, and I think very good morning in Czech Republic. So it is a pleasure for me to meet you all, especially with Alena the first time, although we've been communicating for quite a while, quite some times, through Dr. Muhammad El-Desoki, my colleague at Auburn University in the USA. I met Dr. Miroslava before, so thank you again for coming. We have also some of our colleagues here at SGU. The name, I'm sure you are quite familiar, Swiss-German University, but we are not in Germany or not in Switzerland, but we are in Indonesia. So again, thank you, Alena, for introducing me. Let me just briefly again introduce myself. My name is Kolis Audach. So I work as a senior lecturer and also director for research at Swiss-German University. So it is a pleasure to be here right now. And again, thank you with Miss Clara for arranging this webinar. I was supposed to be visiting Czech Republic in last October or November this year, but due to COVID, we have to cancel our plan. But I think, thanks God, anyhow, we can manage to have this seminar. I don't want to take any credit of this nanoencapsulation. To be honest, I'm new to this area, but it's because I work more on natural products, and I think it will be very good opportunity for us to explore nanoencapsulation technology for drug delivery purposes. So the topic today of my talk is efficacy and toxicity of nanoencapsulation of natural products for drug delivery purpose. Please allow me briefly introduce my university. This is considered as the first international university in Indonesia, established in 2000. The name was attached to the countries involved in the development of the university back in 2000, which is Indonesia, Germany, and Switzerland. So that is the name where the name comes from, Swiss-German University. Yeah, of course, for some people who just learn about SGU, they might think that it is in either Germany or Switzerland, but it is in Indonesia. So we are a non-profit foundation under Indonesian law. We have now 3 faculties or schools. We have 11 bachelor degree program and 3 master degree program. And one of the program is biomedical engineering, where I'm as the lecturer there. We also have other study programs such as food technology, mechatronics, and pharmaceutical engineering, and so on and so forth. So the lecture in Swiss-German University is conducted in English. So as a first, as a mandatory language, the students also learn German as a foreign language for 4 semesters. but don't ask me German. I don't speak German at all. So I just speak in English. We have about 180 full-time faculty members and about 90 part-time faculty members. So if you have any further question about SU, you are free to do so. Okay, before I start, this is my directorate, Directorate of Academic Research and Community Service, which conduct and direct and manage and coordinate all research and community service activities, where we have 4 research centers: Research Center for Food and Health, Research Center for Technology and Innovation, Research Center for Entrepreneurship, Business and Communication, and Research Center for Renewable Energy, Environment and Conservation. Under RCS, we have also Community Service Unit, which also handle the business incubator. So this is something very interesting for us. And also, even though we are just in the very early stage of establishment for the business incubator. So I think I need again to summarize my research, which is actually my journey here, because this is very attached, closely related to what I'm going to talk about, how I started my research and my academic career. I graduated from Indonesian, one of the Indonesian university here in Indonesia, and then I moved to Malaysia in around 97 until 2001, and then I moved to the United States for doing my PhD at Auburn University in Alabama, USA, and then I continue my postdoc training at Yale University School of Medicine in the USA. And then after my departure from USA, I moved to Saudi Arabia. So it's quite worldwide, yeah, to establish a new department in the College of Medicine in one of university in Saudi Arabia, University of Haiel. So I just established a department of biochemistry. I also established laboratory for research and teaching activities. So basically, what I've been doing after my departure from US, I was more involved in consulting work. Yeah, basically, although I work for universities. So this is the summary of my talk today. In the past, I've been working with mainly on molecular mechanism of bacterial defense. Yeah, and currently I work more on drug discovery, mainly on antimicrobial and anti-cancer agents, which is exploring different types of drug candidate sources, especially non-resistant microbial agents from synthetic chemicals. But now most of my work is on exploration of nature products-based medicine from Indonesian biodiversity. The scope of my works, of course, it will deal with efficacy and toxicity of the drug materials, drug target interactions, both experimental and in silico approaches. And I also established Indonesian Natural Products Library, which is a database that has been registered by the Ministry of Law in Indonesia, and the modification of drug candidates, which is now one of them is including nanoencapsulation for drug delivery improvement. And of course, we'll also examine the efficacy and toxicity or safety as well. For the future direction, I will work with, in the very near one, is preclinical studies, which is dealing with pharmacokinetics and pharmacodynamics. So I think that's a summary that will go through it. And I will not go into very detail of my talk because I have a very many slides to share with you. And again, I have to tell you that I'm a new beginner as well in encapsulation, hoping that by applying nanoencapsulation, it will produce a better drug delivery efficiency, efficacy for my natural products based medicines. So, of course, because the title is about nanoencapsulation and drug delivery, I will touch a bit about nanoencapsulation, definition, purpose, techniques, materials, and also drug delivery, factors, drug delivery, and toxicity safety. And also some industrial information from Indonesia on chitosan. Okay, we know that we are facing a lot of issues. Even right now, currently, we are having a very hard time dealing with so many microbial pathogens, including COVID-19s. And in fact, WHO just released not too long ago, 2017, that there are about 12 bacteria for which new antibiotics are urgently needed. And according to WHO studies, that within 20 years, there are about 30 new diseases emerge. So having said all this, it means that the emerging of disease and health issues are becoming harder and harder from day to day. And this is not cannot be competed by the discovery of medicine. So we are really having a very hard time issue. So that is the reason why we are all together need to work to collaborate on how to solve these issues in terms of finding the drugs and diagnostic and also preventive approach. So let me just briefly talk about microbial defense mechanisms, which is that I've been doing for quite a while since my master's degree research. So we have an issue when we talk about microbial defense mechanism. We have an issue with natural mutation. We also have an issue with genes on protein expression as the adaptability ability of microbial to adapt to the environment. We know that, I give you one example, Staphylococcus aureus, for example, which is only 2.8 million nucleotide base pairs in one single day. About 300 mutation takes place in Staphylococcus aureus. So you can imagine we are talking about COVID-19, which is a very small single-stranded RNA, which is pretty much susceptible to environmental change. So that is why it will be very hard from my infectious disease point of view to deal with this virus in terms of finding a new efficient drug and also vaccine. But hopefully we can find one very soon. So the second mechanism of the defense of bacteria in general, microbial in general, is the genes export or protein expressions due to the environmental cues that microbes, in general, they are very adaptive species. They are very adaptive to different types of environmental change, even to antibiotics. That is the reason why there are more and more microbes, such as bacteria, are now resistant to different types of antibiotics. So that is the reason why we are in the very urgent need in exploring different types of sources to find new drugs in different way possibles. So as you can see here, I give a picture here that bacterial flagella, a bacterial, so let me just try to use the pointer. So I have see here. So this is a representative of bacteria. Okay, this is the flagella, the bigger one, the black one, and the little tiny one is the pili. So apparently, this is my work when I did my postdoc. Apparently, bacteria have the ability to express different types of proteins in different types of environment. When they an assembly of pili, when they can assembly of flagella, of biofilm, and so on and so forth. So this is what make us human are having hard time to deal with this bacteria. So we have to find a way to understand the molecular mechanism underlying the bacterial defense. So having said all this, that we are now in the I can say, or we can say, in the desperate situation in terms of how we find the drugs. We have a problem with limited drug discovery. As you all know that the drug discovery process is a very lengthy and expensive process. Even for one single drug, it can reach even until $1 billion US, with the time span 12 to 15 years years. So it is very time consuming, very tedious and expensive, and we have to come up with a way to overcome this issue. So sometimes I've been thinking about this: are we in the losing battle against microorganisms? So of course we should not be despair in dealing with this. We have to do what we have to do, and this is what we are going to do now. Okay, before I start with my natural products, please allow me to briefly discuss, share with you what I've been doing in the past. Yeah, since I returned to Indonesia, pretty much I just started research at SGU. Yeah, because about 5 years ago. So my work started with SU with funding from SGU, seeding funding, and also from a company. So what I'm presenting to right now is my work with the company on establishing drugs from synthetic chemical, which is tetrahydroxyethyl dibisulfite to sodium tests. So it is presumably a peptidoglycan targeting antibacterial agents as a novel non-resistant compound to combat bacteria. Although the title of my talk is about natural products, but I think it will be still open for any other opportunities. In fact, for this chemical, we can try to use nanoencapsulation for these tests. Who knows that we can get a better result in terms of efficacy and also lowering the toxicity of the materials. So we have been testing this tests towards several strains of bacteria. This is the structure of the chemical, tetrahydroxyethyl dibisulfite to sodium. Yeah. So this is actually a chelating agent developed by our company partner, Novis Natura Navita, and has been patented in Indonesia, Australia, and China. So the hypothesis of this is that test is working on the peptidoglycan cell membrane of bacteria by loosen either slightly or totally cut the cross-linking of carboxyl amino bond among peptides in protein or hydroxyl-hydroxyl bond among glycan in the peptidoglycan structure. The unique feature of test as a sulfate chelating agent is the very strong binding formed with the target ligands. So this binding actually the main key of the enlargement of peptidoglycan, allowing the cell membrane to lyse, yeah, so leading to the cell death. So this is again the objective of the work to elucidate the molecular binding mechanism of the test to peptidoglycan bacterial cell membrane. So we have been testing the efficacy of this drug candidate towards several strains of bacteria. The result is quite promising that it is apparently very active towards some of the pathogen bacteria. This is if we test in terms of non-resistant, because we are looking for a non-resistant drug agent, because the current antibiotics are much more resistant, the bacteria are much more resistant to the antibiotics. So we are in need in looking for non-resistant antibacterial. So if you can, as you can see here, in terms of stability, yeah, we see that the test is very much consistent and not showing the resistance towards the bacteria. So it means it's a very promising results. This is the test treated bacteria under microscope. I will explain briefly about the efficacy of the drug candidates toward the bacteria. This is actually showing the picture shows that the cell lies due to the treatment of the test. As we can see in the more detail here, that you can see here that before and after the treatment, you see the size of the cell before is quite large, very large indeed. Yeah, it's compared to the after treatment. So it indicated that the bacterial agent is quite effective in killing the bacteria, both in E. coli and also Staphylococcus aureus. This is also showing the difference in terms of the size of the cell compared between before and after the treatment with the test, about 0.5% test for 35 minutes. Of course, this is just an in vitro study, yeah, although it's a petri dish, we have to evaluate further on in a more physiological condition. Yeah. So again, this is another approach. By using atomic force microscope, we want to see the volume, the size of the cell before and after treatment. As we can see here, that there is a very significant size, yeah, in terms of the size of the bacteria, the cells. If you see here, this is in the micrometer scale in this part, yeah, while here is in the nano scale. So it means that the test is quite efficient towards the bacterial cell. Yeah. So again, it's a very similar result for different treatment, and I will just briefly show you this. And we also did some binding studies using isothermal titration calorimetry. We did this experiment in Malaysia, University of Malaysia Sabah. Since we don't have these instruments here in Indonesia, again, the result shows that the test shows the binding with the peptidoglycan. Although, of course, we need to evaluate further of this binding using different methods such as LC-MS that until now, up to now, unfortunately, we have not done it yet due to some reasons. So, but again, it is a very promising result. So the future direction, studies of membrane interaction using microscopy techniques should be further explored. The fragment analysis of the glycan and test agent to provide deeper understanding of binding mechanism of tests. So again, as I mentioned before, that I'm hoping that I can also apply nanoencapsulation for these tests and evaluate the effect of the nanoencapsulation to the effectivity and also the toxicity of the test towards some bacterial strains. It's our hypothesis. If the test does bind to the peptidoglycan, yeah, then it will have or give different fragments between the treated and the untreated tests against peptidoglycan. So I'm hoping that I can get one or two students to work with this, because it is for me, if I have to do it by myself, it's quite difficult, and it's not easy also for us to find students here at SU. Yeah. So let's move to our second part of my talk, which is the biodiversity-based drug discovery. The reason why I somehow switch from a very fundamental research, which is the mechanism of bacterial defense, where when I did during my master degree, my PhD degree, and also my postdoctoral, is because the nature of the funding later on, I will show you how our government, Indonesian government, really pushed the research towards a more translational research rather than fundamental research. And also is because due to our biodiversity. Yeah, Indonesia is a very well known with biodiversity. So you all know that traditional medicines have been used by the majority of the population all around the world. Yeah, China, India, Indonesia. So I think there's something we don't have to discuss anymore. And in fact, some of very well-known medicines such as quinone for malaria actually discovered in Indonesia a long time ago. Yeah. So although by accident, so it's been a while. And then another drug such as artemisinin, which is for malaria, anti-malaria agent, also come from natural products. But of course, natural products not only in the form or not only in its original form, but also in their derivatives form. Yeah. So for you, I think many of you or some of you who come from Czech Republic, yeah, do not know much about Indonesia. So I think I feel the need to introduce Indonesia in a more detail. So Indonesia is a very rich country in terms of biodiversity. Even we have more than 300 ethnic groups. Yeah, I don't know how many ethnic groups you have in Czech Republic, but Indonesia we have around or more than 300 ethnic groups with different languages.
Only one.
Only one, which is Caucasian. Yeah. So we have about 300 ethnic groups, and each single ethnic groups they have their own what's so-called folk medicine, local wisdoms, if you can say like that. So you can imagine how many local wisdom that we can learn actually from Indonesian ethnic group alone. Not to mention about Indonesian biodiversity in terms of flora and fauna. Indonesia is considered as the top 4 in terms of mega biodiversity, because we are quite rich in terms of vascular plants. There are about 30,000 of vascular plants in Indonesia, in the mix to Indonesia alone. But of course, the plant types or species there are more than 50,000 plants, and we have also actually the majority of fish population in the world. 25% are Indonesia. We also have reptiles, mammals, amphibians, and birds. So quite large number. Not to mention about the biodiversity in marine, sponge and so on and so forth. Coral reef and so on. So we are the sanctuary for different types of bacteria, fungus, parasites, and viruses. Okay, this is again the fact. We are Indonesian population. Okay, we have about 13,480 islands. This is official number from the Maritime and Fisheries Ministry Survey 2010. I think some of the islands disappeared in some period of times, because a long time ago when I started study in elementary school, we know that we have about 17,000 islands. So until now, 2010, we have only about 13,480 islands, which are still a lot. We also have now it's more than 270 million, 2018. And nowadays, 2020, I think we have around 280 million people. So we are the 4th largest country in the world in terms of population. So basically, if you have something, any product, if you sell in Indonesia, you can be very rich. We have a tropical climate. The flora, we have more than 50,000 species of plants, more than 30,000 endemic species. And this is what I need to show you and to introduce to all our colleagues from Czech Republic. There are about 6,000 species used for jamu. Jamu is a type of herbal medicine used by Indonesian people from generation to generation. This is actually what I'm really aiming to explore more in terms of modifying this jamu by using encapsulation and see how the efficacy, the toxicity, or safety of this jamu compared to its original form. Okay, I've been mentioning about the fauna, so we can just skip it. So let me just briefly introduce about the research priority. I think this is because very important also how Indonesian government pushed the research in Indonesia. As you can see here that we have a long-term plan 2015 until 2044. As you see here that natural resources based applied research, not a fundamental research, is the top priority in Indonesia, until at least in the next decade. So this is the reason why all the funding, majority of the funding is allocated for natural resources based applied research. So I think that's something maybe you interest is in your interest as well. Okay, I also would like to introduce how big Indonesian institution is in terms of, for example, number of universities. We have more than 100 public universities. There are about 372, and we have more than 3,000, actually nearly 4,000 universities, private universities. So you can basically pretty much collaborate with anybody here. We have about 4,000 universities and higher institutions. And we have also a very good collection of herbal collection, 2,000 herbal collection in East Java under Ministry of Health. And we have different types of research institutions, both government and industry, that are working on natural product-based research in Indonesia. So let's have a look briefly about what natural products drug discovery. I think this is also quite important, that apparently these are the facts that I think we cannot ignore. About 50% of FDA-approved drug, and also 75% of anti-cancer drugs actually derived from natural products or their derivatives. Yeah. So this is according to a review for about 30 years review. So this is a very strong indication that people in the world, scientists, researcher, industry, are very much looking into the natural products. Yeah. Yeah. You admit it or not, this is just the fact that we have to know. So this is a very interesting fact actually. 75% anti-cancer drugs. Why it happens? Yeah, apparently that United States under NCI, the Natural Cancer Institute, they have this collection. Yeah, they collect about 230,000 unique extracts from extract derived from plant, marine, and microbial organism from all over the world. So that is the reason why it's no surprise to us that 75% of cancer drugs derived from natural product or derivatives. Yeah, so there are some companies. The work is only collecting fractions like Albany Molecular Research in the United States, and also NCI I just mentioned, and some other companies. Some of them already in contact with us, but of course we are having issue also in terms of MTA, Material Transfer Agreement, that now Indonesian government enforce the law for MTA, for transfer agreement. Okay, the topic that I'm going to talk about here is the natural products, of course, but what I'm working on actually on mangroves. So my research now is pretty much since last 5 years ago have been working or dealing with mangroves and their medicinal potential. So again, I didn't know, I was not aware of all these things before, like 5 or 6 years ago, before I joined STU. Before I returned to Indonesia, that mangrove has a very tremendous medicinal potential. Yeah. So as you know that Indonesia has roughly 90,000 kilometer coastline, yeah, the length of Indonesian coastline, and home of about 20 families of mangroves, yeah, and has about 23% of total world mangrove forest. This is the picture that I took in the last survey trips, yeah, where I collected all my samples, yeah, for mangroves. So, and mangroves apparently have a very diverse and highly potential bioactive compounds. So again, here you see Indonesia, almost all entire coastline is inhabited by mangrove. So if we, for example, can move the society to promote, conserve, explore mangroves, it will give a very huge impact to Indonesia as a whole, not only for us scientists, in terms of environmental, social, and economic impact. Of course, people might argue, how about the conservation? Yeah, we will deal with that, of course, because we cannot just simply harvest the mangrove from environment, but we can also deal with synthetic biology and other related issues. Okay, there are so many things that I can collect. I learn, I study from mangroves. At least there are some 8 factors, aspect that I can gather the information from the very early on until now in terms of mangrove research. Yeah, start from sampling, sample packaging, species identification, storage, simplicia, preparation, and phytochemical analysis, extraction, bioassays, and fingerprint. One thing that I really surprised when I tried to start this work, yeah, in terms of developing extract library, not a single institution in Indonesia up to now, yeah, work really seriously in terms of collecting extract. Yeah, so that is I would proudly claim that I was the one who initiated this, the establishment of extract library. We also have consortium, and thanks God, this year, the LIPI, the Indonesian Society, the Indonesian Institute of Science, are planning to build or to establish bank extract, so which is aligned with our research project. So this is just some examples of what we can collect from mangrove research. Species identification. Yeah, this is also something very new to me, that we can always find a new species. Yeah, people might thought that this is species A, but apparently this is not a really species A. So we have to really careful in terms of identifying the species when we come to survey into the jungle or any other places. So we did it in the herbarium of Center for Biological Research in Bogor, Indonesia. This is the certification. So I can say that all of my samples are certified, yeah, by the LIPI, by the Indonesian Institute of Science, in terms of species, because we have to do it, and it's one of the protocol that I established for Indonesian research for biodiversity. So this is one of my favorite picture that I cannot really simply ignore. The reason why is you can see here how beautiful they are with different color. There are orange, there are greenish, and there are brown and any other colors that showing us that mangroves are very rich in terms of phytochemical compounds or components. So again, this is the reason why I am interested in applying nanoencapsulation, yeah, especially with those which are polar, yeah, such as chitosan, for example, because in mangrove there are so many non-polar compounds, yeah, as you can see here. Yeah. There are different types of phytochemical contents. Yeah, we have published this this year only, yeah, recently. So one of my aim currently, my research goal currently is to develop Indonesian Natural Products Library for drug discovery, which is more into database. Yeah, we have registered this, actually already granted the intellectual property for this Indonesian Natural Products Library in 2019, last year. So our aim is to collect, yeah, the definition is collection of natural products containing active compounds used for screening process of target biology. So I am showing here is actually the property of GSK, the one of the giant company, a pharmaceutical company, GlaxoSmithKline. They have these facilities of high throughput screening technologies with the combinatorial chemistry, something that is maybe in the next few decades Indonesia will not have, but luckily Thailand already have these facilities that we are also interested to collaborate with. Last year, the Minister of Thailand, the technology, invited me to give a seminar offline to Thailand. So I'm very fortunate to have that opportunity as well. So the Indonesian Natural Products Library, INPAL, is our database. Yeah, again, this is registered already, granted. So basically, this is the repository link database of Indonesian biodiversity with known function, specifically for drug discovery purpose. The main objectives is, of course, the database, and the second was the repository. Many database, of course, they are not linked to the repository, but what we have here is actually the unique point is the link between database and the repository. So the role of INPAL, a window to Indonesian-based biodiversity research, a platform that links the virtual and physical repository of Indonesian biodiversity. Yeah. So if you see in the United States, USDA, they have a database for every single plant in each state in the United States. So this is actually something that we are trying to cover in INPAL, yeah. But of course, I don't know how long, and also it takes a collaboration with the government. So there are some important features of INPAL. Of course, database. Yeah, there are so many generated data from this research, species identification, geographical locations. In fact, we are now revising our paper. Yeah. That the reviewer asked us to put the geographical location, so we did it, yeah. And also biological assay, of course, methods development, physical collection in terms of extract, fraction, and metabolites. These are generated data that can be collected from a single experiment in biodiversity. Start from species identification, yeah, geographical location, method development, extract yields, and so many other, yeah. Until the biological assay. Yeah, all this data we entry into the database. Yeah. So this is the appearance of our database, although the website or the portal is still under development. Okay, but this is it's gonna be look like. Okay, we have done some research on antibacterial and anti-cancer sources and published some few paper, although the paper is not the best one, of course, yeah, but we are hoping that we can get a better result, a better thorough, comprehensive experiments, so we can publish in a better paper. So this is the one that we have published this year by Antibacterial, yeah. So far, we have about 64 extracts, and 37 out of 64 are apparently active against some bacterial strains. These are some tests, they are bacterial tests, and if you see here, the result is quite promising, especially both for E. coli and Staphylococcus aureus. That is the reason why we further study about these two bacteria. And this is also a very promising results. Following this Staphylococcus aureus test, we did also test it against MRSA, which is one of the most prevalent pathogens nowadays in Indonesian hospitals. Yeah, there is a very promising result for MRSA, anti-MRSA. This is the one that we are now revising the paper for a journal. Yeah. About the cancer activity. So from our studies indicated that mangrove has a very good potential as anti-cancer. So it will be very interesting if we can see this, modify all these extracts using nanoencapsulation. So finally, we come to our main topics, the nanoencapsulation and drug delivery. Please don't expect a very high technology or whatever it is, because again, I mentioned before that I'm also started to learning about these topics, the nanoencapsulation. Although I learned this about maybe 6 or 7 years ago about nanoencapsulation, but didn't really have a chance to deal with this. Although our collaborator, I think also Dr. Siswanto, also present today, this afternoon. Actually, he is my lifetime mentor. Yeah, he is my undergraduate advisor, and until now, I'm still learning from him about many things in terms of research. He is very active, although actually he is supposed to be retired, but the institution that where he works still need him, so he's still active doing research, and I hope he can join later on. And he also asked me to present some of his work in nanochitosan encapsulation. So this is actually the some of the coverage topics that I will cover. Yeah. So I hope that I'm not salting seawater. Yeah, but since there are some of you here are my students that are coming to attend the lecture, so I think I need to start with definition about encapsulation. So encapsulation relates to technologies which enable to formulate one active compound or more. Okay, so it could be consist of one or more active compound in one matrix inside individualized particles with a specific geometry and properties. Yeah. So microencapsulation, of course, dealing with the size in the micro size, while nanoencapsulation, of course, the size is in the nano size. Yeah, even sometimes more than a few micrometers. So it's still considered as nanoencapsulation. So nanoencapsulation is defined as the process of encapsulating substances with various coating materials at the nanoscale range. So this is the structure of the matrix. If you see here, it's simple. We have this very simple but powerful picture. How to describe, illustrate the structure of the matrix of nanoencapsulation. So this is actually the encapsulated materials, which mainly the active compound, yeah, the active substances, yeah. And this is the shell, and this is the thickness of the shell. So there are two types of matrix structure. One is dispersed. Yeah, dispersed. It's not really joined together, but dispersed in the entire core. Yeah, and the second one is joined together. So it really depends on the properties of the compounds, of course, the active compounds, and also the shell itself. Okay, so the matrix structure is dispersion of active compounds in the encapsulation materials. It could be beads, micro or nanoparticles, microsphere, yeah, which are the actives is in orange. Yeah, while in the second matrix is the core or cell structure. The active compound, pure or not, is confined as one core by one shell or more, or multi-cell. It could be multi-cell, not only one shell, or micro or nanoparticles, micro or nanoparticles, or microcapsules or nanocapsules. Yeah, but we could also combine all these two matrix. Yeah, where the matrix core or shell, or core or matrix shell, two compounds or more, one in the core and another or one in the shell. So it could be mixed both in the core and also in the shell. We can do that also. Yeah, it really depends on the properties of both matrix, the shell, and also the core itself, the active compounds. So I will just go briefly about the component of encapsulated materials. Yeah, the active compound could be physical states, could be solid, liquid, gas, could be also hydrophilic or hydrophobics. This will pretty much determine what types of matrix or the encapsulation that we are going to use. If we have, for example, we have nature product, which are mainly hydrophobic. So how we try to encapsulate these hydrophobic substances to go through, for example, solution, because we know that the hydrophobic compounds will not be able to solve in aqueous solution. So we have to somehow solve, or encapsulate this compound with something hydrophilic, such as charge like chitosan, or vice versa. If we have, for example, hydrophilic compounds, then we want this compound to enter the membrane, as we know that the membrane are very much insoluble, because lipid bilayer. So we have to encapsulate these hydrophilic compounds with something more hydrophobic, such as liposome, for example. And also, it will also depend on granulometry. Active compound contain up to 900 milligram per gram of final particle. So if you see here, the mass actually quite massive. It can load to or occupied 900 milligram, which is quite a lot of mass actually, yeah. And also depends on the appropriate grade cell be selected, either for food grade, feed, cosmetics, pharma, and others, yeah. Because nanoencapsulation actually mostly for three area: food, cosmetic, and pharma. Because for feed, it will be very much more expensive, yeah, to feed the poultry, for example, using nanomaterials. While the encapsulation materials, which is the shell, it could be mainly biopolymers. Yeah, it could be derived from vegetable, animal, or bacterial sources. Yeah, such as alginate, pectin, chitosans, yeah, carrageenans, Arabic gums, and other. So there are many different types of biopolymers. The one that I'm going to talk about more is actually chitosans. Yeah, in fact, Dr. Siswanto asked me to present his research in chitosan, in nanochitosans. The encapsulation can be also wax and fats that I just mentioned. It could come from vegetables, animal sources, yeah, and there are different types of wax or fats that can be used for encapsulation materials. Surfactants also can be used for encapsulation materials. This is good for example, for drug delivery in the membrane, inner membrane. So the materials that we are encapsulated can enter into the membrane. And also there are synthetic polymers, yeah, PVA, PEG, polycaprolactone, and other. There are so many polymers. Yeah, the thing is just later on we have to deal with how all these polymer encapsulation materials eventually not toxic to the cell. Yeah, because that is the ultimate goal of the encapsulation. Yeah. If we, for example, in one way, we will be able to increase the efficiency, but it will be much less useful if at the end the product will be more toxic to the cell. Okay, as I mentioned, the primary use for nanoencapsulation is for pharmaceutical, because they are expensive, food grade, and also cosmetics, So only for expensive expensive materials, okay? Not only because the cost will increase significantly when we try to, for example, chitosan. If we want to upgrade to nanochitosan, then we have to do further development, using chemical or any other methods. Okay, so what is actually the main purpose of nanoencapsulation? Of course, in terms of pharmaceutical, we want to improve bioavailability. By improving bioavailability, then our cell can absorb more the active compound, also solubility, stability also, because we want our drugs stable. So one way to do it is by encapsulation. Physiological performance, we want our drug to be more physiological function, by hoping that by doing encapsulation it will improve also. Also, in terms of drugs and also foods, encapsulation or nanoencapsulation can also improve organoleptic properties such as taste, smell, and other related things. And also functional characteristic. We want to improve a functional characteristic of our active compound. Of course, the reason why nanoencapsulation is quite now one of the very emerging area of study, research, and even in industry, because there are different or various advantages of using nanoencapsulations. The nanoencapsulation are the ultra-thin layers should significantly improve mass transport. The smaller, of course, it will be more efficient in terms of transport. And also the volume, the reduce the volume. Its ability to protect active pharmaceutical ingredients. This is one of the main or even the most important purpose of or function of the nanoencapsulation, that we want to protect our active pharmaceutical ingredients from degradation. The next is to improve the precision of drug delivery targets by utilizing surface coating or conjugating that ensures adequate cell entry. It depends on the properties or the nature of the active compound. Then we use different types of encapsulation. The idea is how our active compound reach the cell where we target as the target of the drug. Nanoencapsulated drugs can be labeled with fluorescent probes for imaging purpose, which is particularly useful for evaluating drug efficiency during preclinical and clinical studies. This is quite important. So we want to see whether our drug is effective or not. Or for example, if we want to do some preclinical studies, we use, let's say, zebrafish. I hope Dr. Intan, Prof. Intan from UPM also attend the seminar. I haven't seen her name just now. So by using this, we can, zebrafish is quite transparent. We can see this, if we can label the nanoencapsulated by using fluorescence, so we can track the drugs, where the drugs going from one place to another, and how our body or animal model use the drugs efficiently or not. Okay, so it's really very important aspect to work with. Nanoencapsulation is considered to enhance availability. Of course, this is one of the very most important aspect, that is we want to improve or enhance the availability through improved uptake at cellular surfaces. The smaller size of nanoparticles means that they will remain in suspension longer and scatter less light than larger colloidal particles, so allowing use in a wider range of transparent aqueous foods and drinks. So let's briefly talk about efficacy of nanoencapsulation. The efficacy of many nanoencapsulated and microencapsulated process often relies on uniformity in particle size and distributions. So one of the reasons why nanoencapsulation is quite effective is because it will be very good, for example, that in terms of uniformity, that we reach our, let's say, homogeneity of uniformity to the highest extent. to improve the efficacy of the nano-encapsulated drug. Yeah, so it really depends on the uniformity. I think with certain methods we can also always improve the uniformity of our particles. So the use of supercritical carbon dioxide has shown promising benefits to ensure the optimal design of particle size in both microencapsulated and nano-encapsulated drugs, as well as the ability to control drug loading process at various temperature, pressure, and flow ratio range. Okay, for those who haven't learned about drug loading is the process of incorporation of the drug into a polymer matrix or capsule, which is one of the very important process in encapsulation process. And also drug release is the reverse process by which the drug molecules are liberated from the solid phase and become available for absorption and pharmacological action. So these two things, the drug loading and drug release, will determine the efficacy of the nano-encapsulation that we are establishing. Okay, the factors affecting nano-encapsulation methods of choices. There are many other factors. Of course, the nature of drug and encapsulating polymers. It really depends on the nature of drugs and also the encapsulating polymers and how we want to make it. As I mentioned before, if it is non-polar, then we have to make encapsulate with polymer which are polars and vice versa. The final destination is, or the final objective is, in how our bioactive reach to the target. So the desired properties of the particles of the drug and the encapsulating polymers that have to be considered, there are size and size distributions, morphology, drug loading, release kinetics, residual toxicity, solubility, mutual compatibility, and also stability under proposed experimental conditions. So one important thing to note is that we have to consider very carefully about the stability of our nano-encapsulation under proposed experimental conditions. So it really, again, we need to adjust this to the microenvironment, even during the process making and also during the application. It is of great importance to achieve the highest possible encapsulation efficiency, meaning that loss of drug during encapsulation procedures has to be minimized by careful choice of experimental conditions. So therefore, it is important to verify that proposed encapsulation procedure does not affect pharmacological activity and of the encapsulated active. Okay, I will talk briefly about the next encapsulating techniques. So in general, there are two techniques, which is top-down and bottom-up. And some researchers believe that top-down is the better compared to bottom-ups, because top-down we can really design. Top-down is when top-down approach is utilized, precise tools are used to accurately reduce the size and structure of the materials. So the reason why top-down is better compared to bottom-up is because we are in more control in terms of designing the size and the structure of the materials compared to bottom-up. Because in bottom-up approach involves the use of materials derived from either self-assembly or self-organization molecular process. So it is more a random process compared to the top-down one. So these are some trends, recent technologies in nano-encapsulation. There is nano-emulsification, nanopolysomes. This is actually, nano-liposomes. This is actually the advancement of the liposomes technology. They convert the liposome into nanoliposome as well in the chitosan. They improve from chitosan only to microchitosan to nanochitosan. There's also method electrospray, nanostructure formation through the use of cyclodextrin, solid lipid nanoparticles, and also nanostructured lipid carriers. I'm not going to go into detail of these technologies. I just briefly mention about the technologies in general. So this is one of the very famous, actually, one type of nano-encapsulation, which is the liposome, which is now improved to the nanoliposomes, actually by employing the characteristic of lipid bilayer. For you guys, students, we learn about lipid bilayer, membrane, phospholipid bilayer. We know that lipids has two different parts, in their structure. The first is, of course, the polar head, which is where the carboxylic acid is, and the second is the non-polar tail, where the aliphatic chains of fatty acids. So by combining this, utilizing the structure of the lipids, by employing the structure of lipids, we can use this bilayers and use for encapsulation of different types of active compounds. So if you see here, mainly I can guess that if you, for example, the liposome is actually made for encapsulation of polar active compounds. Polar actives, polar or soluble actives compounds. Because we use non-polar or hydrophobic encapsulation. So the idea is to bring, to deliver the drugs in the form which is hydrophilic or have charge or soluble in water to pass through the membrane. Okay. So this is the encapsulation process. The first is incorporation of the active ingredients within the microparticles or nanoparticles, the dispersions, liquid core, or agitation and spraying if it is a solid core, and also the stabilization of droplets, liquid core, or film formation if it is a solid core. So these are, in general, there are three principles of encapsulation process. Incorporation, dispersion, and also the stabilization. Okay, now comes to the application of nano encapsulation. How much time I have, Alena? I think it's already 1 hour. I will finish in a few minutes.
Thank you. 5 minutes time.
All right. So we know that the main purpose of nano encapsulation application is to reduce transport and storage costs through the production of powders, to protect components from deterioration and storage, to partially protect or target delivery during digestion, to disguise unpleasant tastes and odors, and to improve the dispersion of fat-soluble matters in aqueous medium and enhance their bioavailability. So the main reason nanoencapsulation is so widely used is its efficiency in protecting the core material and ultimately releasing the API or the active ingredients when required, the active pharmaceutical ingredients when required. So in the context of drug delivery, this is why nanoencapsulation and nanoencapsulation is one of the most promising method in drug delivery. So some of the most common application of nanoencapsulation, therefore, can be found in a targeted drug delivery system that will only release drug upon its arrival at the correct location within the body. Time-release drug delivery system, in which slow degradation of the external phase allows for controlled release in the body. So this is actually one example of type of drug delivery system that can be found in nasal drug delivery devices. And then embedded fragrances for perfume garments, food additions and enhancements, increase shelf life and stability of over-the-counter pharmaceutical products such as vitamins. So I think this will also be very good for, I think people or scientists need to try this for vaccine, COVID-19 vaccine. I think someone need to discover the efficiency of the vaccine, which is until now cannot be released yet for due to some reasons. So this is the last part of my talk, which is the application of the nanochitosan. As you know that chitosan is derived from actually the animal's shell, such as crabs, such as shrimp, the hard shell of those animals. This is the structure of chitosans. And now people make nanochitosans, and apparently nanochitosan have more effectiveness, more efficacy in terms of efficacy due to the size. So the method, this is strictly from Dr. Siswanto Group's works. So they were able to synthesize the nanochitosan from chitosan by metal, which is copper. So the synthesis of nanochitosan and nanochitosan copper was carried out by ionic gelation methods. So they were able to reduce the size of the chitosan from 606.5 nanometer to later 386.8 nanometer, and then reduce again to 254.1 nanometer by using nanochitosan copper. So it has been used for antifungal, and it's become very effective. As you see here, the percentage of inhibition of chitosan, nanochitosan and nanochitosan copper by 35.19%, 49.49%, and eventually 100% respectively. So you see, if it is 100% already, it means there is no further development required. We just need to be aware of whether this will affect the cell or not in terms of toxicity. Because if we, for example, try to reduce the size of the particles, we also have to take into consideration the toxicity of these particles that might be enter different types of cell, which are not necessary, and in fact maybe could be harmful to the cells. So those between advantages and disadvantages, or the benefits and risks and benefits, always need to be taken into consideration. So these are some examples of application of nano-encapsulation of natural products. Nanosystems for the encapsulation of natural products, the case of chitosan biopolymer S matrix, and also these some articles, supramolecular nanoencapsulation of anabasine reduced its developmental toxicity in zebrafish, and some other paper that I have tried just to share with you. Okay, a little bit about drug absorptions. We know that our talk about today is about drug delivery. It means that how our particles, the nanoencapsulation particles, would affect the absorption. So we have to know a bit about different types of absorption mechanisms. Of course, we are not going into detail, but my point is that we just have to consider different aspects when we try to make nanoparticles, because our nanoparticles might be absorbed at the wrong place and at the wrong time due to unprecedented event. For example, the point is that we have to be careful about taking into consideration different aspects when we talk about drug absorption mechanism for nanoparticles. So because we don't want our drugs absorbed in the wrong place in the wrong time, so it eventually not only not cure the disease but damage the cells. Toxicity of nanoparticles, toxicity assessment or understanding the complexity of toxicity of nanoparticles products is required to understand possible adverse effects and their fate inside the human body, which include but not limited to physiological impact imparted on cells. Of course, we have to know the potential physiological impact to the cell, also different physicochemical properties of nanomaterials. We hope to consider the size, morphology, because this is also very important for the cell proliferation, for example. Major pathways of nanomaterial uptake in the cells, we also have to consider this. That is why pharmacokinetics, pharmacodynamics studies have to be done as well. Possible methods for human exposure to nanomaterials, for example, skin, respiratory tract, gastrointestinal tract, blood-brain barrier, liver, and spleen. And also types of assays. We have to do some assays in terms of cytotoxicity, cell proliferation, and genotoxicity assays. So there are many different aspects that we have to consider. So this is the future direction of my work, which is hopefully very close, because I am just submitted a proposal to the government. So by the way, my research has been funded in the last 3 years by Indonesian governments. The first year I got funded from SGU, and then later on by a company, and then by the Ministry of Research. And hopefully for the next 3 years I get the funding to do all this work. So in conclusion, acceleration and effectiveness of drug discovery is required to overcome the emerging of disease. Both synthetic and natural products need to be explored as drug resources. Natural products-based medicine from Indonesian biodiversity opens wide selection of drug candidates. Mangrove extracts are potential as antibacterial agents, according to inhibition study with considerable activity and can be used as good candidates for nanoencapsulation. Nanoencapsulation of natural products for drug vendors are promising methods in enhancing drug delivery. Toxicity or safety of nanoencapsulation or nanoparticles need to be taken into consideration when applying the products for drug delivery purpose. And last but not least, collaboration is a must in order to strengthen our efforts for drug discovery process. So by the end, I would like to thank all my collaborators, Dr. Aina Sefku from TUL, Dr. Mirka, and also Dr. Mohammed, and also from funding, from the ministry, from the companies, and also from SU, of course, some of my former students, and also some of my collaborators in different institutions, UPM, UTM, MS, IPB, and now I have a new collaborator from TUL Liberic, Czech Republic. Thank you again for your attention. If I may, Alena, I have just need to present of my, actually we can also discuss this later after our seminar, but if you will allow me to just briefly introduce the work of Dr. Siswanto, because he asked me to present this, and I think also it's very important. Is that okay?
Well, of course. Maybe if you don't mind, now I will start the discussion. Thank you very much for your interesting presentation.
Okay,
I will discussion for those who need to leave, maybe to ask you questions about your presentation, and then we can follow with the next presentation on the chitosan. Okay.
All right. Thank you.
Okay. So thank you very much again, everyone, for attention. Now it's time for a few questions. Maybe I will start with one. You have already the extract library from mangroves, so I believe you have many samples. Do you have any plan for the prioritization of the drugs of the extracts to be used for further studies?
Yes, we have some that we are now working on that has been tested against MRSA and also cancer cells. So those two extracts that we are going to be used for further studies. Especially, I'm looking forward for the encapsulation with you guys. Because in the next 3 years, I'm going to do some clinical studies as well. So I think we need to test this again using nanoencapsulation.
I think to work further on the extract libraries, not just mangroves, but also other plants.
Yes. Okay, let me just briefly mention again about the database, because the database actually will be open for any researcher who is using biodiversity from Indonesia. But of course, I cannot do it by myself. So it will be like, that NCBI, that people can deposit their data through the website. But this is particularly for Indonesian biodiversity. So, but for time being, from my side, I will just stick to mangrove, because even only mangrove alone, I cannot handle it. It's too many to handle. Yeah. But of course, we're working on our colleagues here working with curcumins. I know in Europe, curcumin is very famous for cancer treatment as well. I think our collaborator in IPB University work with curcumin for cancer.
Can I have a question?
Sure, yeah.
Thank you. Thank you for your lecture, colleagues. I'm just curious about your natural extracts stability in terms of thermal conditions and time conditions in the storage.
Yes. So we tested our stability. Yes, it is not stable. Yeah. So within 6 months or 1 year, it reduced about 10% activity. So that's one thing that we already written for publication. Yes, it is not stable. So I think that will be good direction for us if we can use encapsulation to stabilize our extract, right? So this is one technology to use for stability of the extracts.
Okay, wonderful.
Yes. Thank you. Because there are some, that when we extract some things, there are some volatile compounds that might release right away or within 1 week or 2 weeks. But within 6 months, I think I would say a year, it reduced about 10% activity. Yes, it is not stable. Any other question? It's very great to see Dr. Mohammed.
Okay, thank you so much. It's a great pleasure.
20 years, 18 years, yeah?
Yeah, around 18 years. I see the gray hair on your face. So yeah. And I would like also to welcome the biodiversity in this webinar today, like the people from Indonesia and from other countries, which is very rare in our webinars in Liberic, to find this biodiversity also in the webinars. So it's a great opportunity to have you here today and all the people from Indonesia today also. And I would like to just make a comment about the extracts that these extracts and the waste, actually the waste that comes out of these materials. Can we identify them? Of course, this can be in further discussions, but briefly, what I'm thinking of is using the materials or the waste of these materials to utilize them in other products or other things that can be useful as well. So I'm not sure what kind of polymeric materials that come out of this special plant and special material that you have.
Yes. Basically, what we are trying to do is we are trying to have zero waste because basically we can pretty much extract anything, right? It depends on what type of compounds that you want to obtain. Let's say if you want to get more polar compounds, then you use a polar extracts and so on. Solvent. If you want to get a more non-polar compounds, then you use non-polar solvents. But then you're right. There are many things. I give you one example. Because in the plant there are also enzymes. There are many enzymes, incorporated into the plants, either in the roots, in the leaves, and so on. But one very famous waste is actually palm oil waste. If you utilize palm oil waste, actually you can be a very rich man because palm oil is very rich in lipase. Palm oil also rich in amylase, so you can use either for poultry as a feed or maybe for industrial purpose. Of course, if you want to use for pharmaceutical grade, you have to go a little bit longer in terms of purification, isolation, and so on and so forth. So there are different types of waste. One important thing is actually in the world now, actually mangrove associated also with microbes. There are research in mangrove microbes. People try to find antibiotics, different types of antibiotics from different types of microbes associated with mangroves. Actually, in 2015, there was a group from Germany, who traveled around Indonesia to collect actinomycetes. Actually the soil of mangroves. Actually, the soils are very rich in microbes. And if you, I can mention the AAD have been working long time, specializing in biodiversity. In fact, they have funding for biodiversity research in different countries. Because, well, I don't know about Czech Republic. Are you guys interested or not in biodiversity research? But Germany is one country that is very interested in working with this biodiversity. Not to mention about marine, there are so many others. So mangrove has plenty of microbes that people are now working on to isolate the microbes to get the antibiotics either from fungus or from bacteria. Thank you.
Yeah, thank you.
Yeah,
thank you. And by waste, I may refer to the wrong thing. Maybe it should be termed as side product, like because the main objective of your extract to be like for pharmaceutical. So maybe we can name it the side products of the same.
Yeah. Right. We can. We just need to know different types of mangrove. Yeah. When we talk about mangrove, of course, in the mangrove context, we have to be careful as well because there are some toxic mangroves. So mangroves not only good, but also there are some toxic mangroves. Yeah. So the waste can be used for different types. Yeah. We can use for feedstock. Yeah. If we want to use for feedstock, maybe for enzymatic analysis. Maybe if you want to, let's say, isolate some enzymes from the mangroves. Yeah, I believe there are many other things can be used or can be done from mangroves. Just like any other plants, I think.
Okay, thank you. Maybe one last question.
Okay.
If not, then I would like to thank you all, and we will continue with this chitosan part. Okay, maybe
all right.
Smaller, but of course, anyone who is interested can stay. Thank you.
Okay, thank you, Helena. Yeah. So I shall continue. All right. Actually, this is more into industrial part of the work because our collaborator, Dr. Susanto, maybe also he's here. He can help also later on. So let me just switch the presentation. Actually, I asked him to present himself, but he asked me to present it instead. But he will join us later in the discussion. Okay. So, anyone can see? Hello?
It's uploading.
Yeah. Okay.
Okay. This is the current research of our collaborators, Dr. Siswanto Group from Indonesian Research Institute for Biotechnology and Bioindustry. He gave me this presentation, current research on chitosan from crab shells for today's seminar. So the potential production of chitosan in Indonesia from waste of shrimp shells and crab shells in 2018, as many as 165,120 tons per year. So potential 24,700 tons of chitosan per year. So this is a quite huge number. Yeah, I don't know how much the world required chitosan per year. So total potential production of chitosan in Indonesia is about 29,800 tons per year. This is the production process of chitosan without heat from crab shells. Yeah. So I will just very briefly, yeah, not very, because this is, I think it's more technical. So how you make nanochitosan from chitosan, yeah. There is a pretreatment, demineralization, deproteinization, deacetylation, yeah. And yes, you can see every step. As you can see here, there will be a waste that can be utilized. You see the deproteinization, it means it will leave a lot of proteins to be utilized. So the idea is zero waste, yeah, from HCl to sodium hydroxide for precipitation of calcium protein sodium chloride plus water. So the production cost, using heat process is 460,000 rupiah per kilogram, which is, okay, let me just calculate. 460,000, about $30 per kilogram, roughly. Yeah, don't be surprised. We have many zero. Production costs without heat is about less than $20. Yeah, about $15. Yeah. And the selling price is 700 to 900,000 Indonesian rupiah per kilogram. Yeah, so it's about $60 per kilogram. So this is the pretreatment of crab shells. Yeah, we are, of course, 70% of our maybe more than 70% of our area is ocean. Yeah, Indonesia is plenty of islands, so we have plenty of crab shells. Yeah, or maybe also shrimps. This is the prototype equipment for chitosan production. It's quite modernized. Yeah, and then the production of chitin and chitosan from crab shells and shrimp shells, because chitosan is actually the derivation of chitin. Yeah, this is the raw material, the crab shell. This is the chitin and also the chitosan. While from the shrimp, yeah, this is also, I think the shrimp will be less than the crabs, because the shrimp is shell is a lot thinner than crab shells. Okay, the chitosan quality test results. I cannot really explain much about this because it's and this is the potential markets for chitosan, bioran, seed treatment for seed for agriculture. Yeah, the agriculture for wastewater treatment. I think Alina will be interested in wastewater, I believe. Yeah, or agriculture, or maybe Mirka will be interested in for foods or medical purpose. Yeah. So this is the bioassay of copper chitosan against Colitrium, the fungus that I mentioned about. Yeah, that is effective, 100% effective against fungus, which is quite impressive to me. It will be very interesting for me to test as well using our extracts and see how these copper nanoparticles enhance the efficacy of the extracts, yeah, towards different types of bacteria or cancer cells. Yeah. Okay, this is the bioassay test result of chitosan, nanochitosan, and copper chitosan that I mentioned in the presentation with different percentage, yeah, up to 100%, yeah, 100% with copper chitosan, which is very impressive. So this is again about some of the product industrialization of chitosans, yeah, using 700 liters of Industim. In Bogor. Bogor is an area not far from Jakarta. This is farmers' demonstration plot for Industim, which is the product. Industim is the product. And this is farmers, they're using different chitosan product for agriculture. Yeah. And this is for onion and chili for agriculture. And that's pretty much about the application of chitosan for agriculture mainly, because Dr. Siswanto is actually working more in the estate crops biotechnology. Yeah. So I forgot to mention that Dr. Siswanto graduated from Montpellier University in France, I believe, Pak Siswanto, yeah, in biochemistry a long time ago. So he is more in agriculture, while myself is more into biomedical area. So I think that is enough for this presentation. Maybe Dr. Siswanto could add some more if you like, and if you the audience want to ask some question, feel free to do so. But of course, I will not answer the question because Dr. Siswanto is more appropriate to answer the questions. Yeah, but I can help to explain. Okay, Alina, that's from me.
Okay, thank you. So maybe, as you said, if Dr. Estananto would like to add something.
Yeah, Pak Siswanto, are you here? Pak Dr. Siswanto? Yeah, I couldn't hear you. Hello?
Yeah, the microphone is still turned off.
Yeah.
I'm not sure.
Yeah. Okay, I can hear. Pasis bisa ini? Oh, I think he's having problem with his microphone, yeah? I can see him, but and he's trying to say something. Nggak kedengeran Pak Sis? Oh, very unfortunate, yeah. So let me see the chat room. Hello, Pak Sis. Can you hear me? Oh, okay. Then
I think he can hear you, but he doesn't. Like, the microphone isn't working.
There's something wrong with his microphone, yeah?
Yeah, but it's a good opportunity to see him at least,
Yes,
and listen to.
I mentioned he's my lifetime mentor.
Exactly.
My role model in doing research, yeah. Well, now, even though my hair is whiter than his. So I'm I'm really sorry that I couldn't make it to visit Liberic, Alena, Mohammed. But hopefully next year it will be better, the situation. And thank you for Mohammed for introducing me to Alena and also Mirka, you know. So it's a very good opportunity for me. Yeah.
Thank you. And it's a great pleasure. And by the way, now I'm talking to you from Switzerland. So yes. And yeah. And actually, my group here also they have a lot of work with chitosan. They are working a lot with these materials in general, hydrogels in general. So there will be a lot of possibilities. I hope to to benefit of both sides from this kind of common or shared research. So.
Okay, great. So at least if you need some chitosan, just come to us.
Definitely, yes. Yes.
Well, what are you doing, Alena? What are you working on right now? I know you are more into environment. Am I right?
Yeah, it's but it's as I introduced before. It's nanotoxicology. New course also here. I'm not sure if she's still present or not. So it's nanoecotoxicology field.
Right.
Now we started to work also with microplastics and the fate in the environment and the effect on the bacteria.
Right.
Another field is also, let's say, photocatalytic surfaces for self-cleaning or for destruction of bacteria and algae on the facades of buildings or even like sculptures or some historical monuments. My colleagues started to collaborate with some Indian group and with their application on this. So I would say that I don't focus on just one thing, but in my department there are groups that are focusing on different things related to microbes or microorganisms, and Mirka is here presenting the group that deals with more the medicine and the development of the different scaffolds or materials for medical use.
Okay, very interesting. It's quite diverse.
Yeah, it is diverse. So the thing is that most of our research is applied, but we also aim to work more in fundamental research. And but applied research is very interesting, and this chitosan stuff and nanoencapsulation of the drugs, natural drugs, is definitely very interesting. But it may be more on Mirka. I'm not sure if she's still here, so maybe she can add something. Hi, Mirka.
So
I think Mirka, you probably also worked with chitosan and
yes, exactly. Yeah, actually, one of our projects is focused on development of chitosan-based nanofibers for contaminated wound treatment. Because as you know, chitosan has plenty of benefits even for this application. And yes, we already did some work with natural-based products in terms of pharmaceutics with some extracts, for example, containing thymol, we mentioned in your lecture. And the results were pretty impressive, actually. We plan to publish the paper on this topic next year. So hopefully it will work. So I think there's many topics in common. We could be cooperating and help each other, I believe.
Sure,
as you said, it's a pity that we cannot meet face to face, and it's not so easy to talk through just the monitors. But anyway, it was really interesting and a great pleasure to see your.
Dr. Sifanto pretty much work on the water treatment, something like that, So it's all very important. So anything related to bacteria, I can help.
Okay. It would be really great if you can visit us next year.
Sure, yeah. So I'm looking forward to it.
Yeah, as we propose, the best way would be to collaborate through student exchange or short visits and develop some small projects leading to some public meetings.
We have Dr. Dedi here, maybe our head of the department. Maybe he can add something regarding the student exchange, because of course, SU as an international university, every year we send our students, around 200 students, to Germany. So I think it will be good also if we can diverse our country's destination. Last year, one of our students did internship at Basel University at Switzerland, I believe, Basel in Switzerland. So, Pak Dedi, if you have something to say, maybe, for student exchange?
Yeah, sure. Usually after our students need to do second internship in abroad, usually, and if they take double degree, and so after about 1 month having an intensive course at Germany, they are free to go anywhere. Usually they pick place in Germany, but sometimes they can also pick other places in Europe. So I think it would be nice also if TU Liberic could be an option for them also to do their internship, and then if they can go at your place, then they could have we can have collaboration to do that small project together, for example. And recently they also some of our students went to Universitybased also to do some internship there and conduct some small research project. And for about 6 months as internship student, and not bad. Sometimes they can produce also some research paper, maybe at least at the conference, but sometimes also they can produce also full research paper for journal. One of our students that did internship in KIT, Karlsruhe Institute of Technology, also wrote a paper based on her internship that time. So yes, we could explore that possibility, and maybe we will work together with our internship department, and maybe when we can have an MOU, for example, for this exchange student, then we can make TU Liberic as one of our preferred destination for our students when they do their internship abroad.
Yeah, okay.
Yeah, definitely, and I would like also to extend that, now here at IMPA in Switzerland, it's like part of the ETH domain. So we have here students from ETH Zurich and EPFL and all the technical universities in Switzerland who are actually doing their internship and this short study period, as you mentioned. So mainly it will be a good opportunity for them to go to Liberic in Czech Republic. But I'd like just to make this also a possibility. If I can help in this kind of effort, I will be glad to do that. Thank you.
So, thank you very much. So we have the more opportunities, the better, right? Yeah. I just so far couldn't send them to US. It's not easy. Yeah.
But Auburn is open also. Auburn is, Auburn, how people there are. Yes.
Yeah, I miss Auburn.
Yes, both of us miss it.
Yeah. So,
by the way, do you know that Dr. Al-Mughazi?
Yes.
Professor Mughazi. Yeah, he passed away last week. So he died last week. Yeah. So
last week only.
Yes.
Okay.
Yes.
I'm sorry to hear that. Yeah, he is a very expert in statistics. I have to remember him. Yeah. Okay. Any other? Maybe how can we proceed this, Alena?